Hello everyone, my name is Oceane Campion and I'm really grateful for this opportunity to present my work about LRP1 matricellular receptor involvement in triple-negative breast cancer tumor angiogenesis. So my research focuses on triple-negative breast cancer. As you know, about 15 to 20% of breast cancer are found to be triple-negative. It's a rapidly developing cancer with a high metastatic potential, where the cancer cells are tested negative for estrogen receptors, progesterone receptors, and HER2 receptors. In the absence of these markers, TNBC patients do not benefit from currently available receptor-targeted systemic therapies, such as hormonal and trastuzumab-based therapies. Thus, TNBC remaining refractory to targeted and conventional therapies advancement demands identification of novel therapeutic targets to enhance therapeutic options. So in my research team, we have concentrated our efforts on the multifunctional LRP1 receptor, standing for low-density lipoprotein receptor-related protein 1. So when we say multiple function, inevitably it means multiple levels of influence. And that's why its involvement as a pro or anti-tumor receptor appears controversial regarding the type of cancer. Concerning breast cancer, a previous anatomopathological study revealed that LRP1 expression was correlated with TNBC and HER2 subtypes. About angiogenesis, LRP1 regulates proteolytic cascades and EMC remodeling, which are necessary for angiogenesis. Moreover, LRP1 has emerged to be involved in the regulation of physiological vascular homeostasis in various vascular cells. So this prompts us to take an interest in its involvement in the TNBC progression, and especially to its role in controlling the angiogenic balance and whether this role could be decisive for tumor progression. So this is the subject of a recent publication of mine that I'm going to talk about. So the first objective focuses on the tumorigenic potential of LRP1 and its influence on tumor angiogenesis in vivo. And the second objective led us to evaluate the influence of LRP1 on the angiogenic secretome of tumor cells in vitro. So the technique chosen and developed to investigate LRP1 function was the use of RNA interference. So to obtain a TNBC cellular model, MDA-MB231 cells were transfected either by a plasmid containing an shRNA directed against LRP1, leading to what we call shLRP1 cells, or the same plasmid, but designed to express an shRNA without target, which constitutes O control, leading to what we call SH control cells. So we validated the repression of LRP1 expression in this model at the transcriptional and protein level, with an average of 42% and 58% decrease, respectively. So to confirm the involvement of LRP1 during tumor progression in vivo, we used an orthotopic xenograft immunosuppressed mouse model. So we injected 5 million of SH control or shLRP1 cells into the mammary fat pad of the mice, and we followed up the tumor growth over 28 days. As you can see, we measured a tumor volume decrease of 64% in shLRP1 tumors compared to SH control. And the count of mitosis did not reveal any difference, suggesting that angiogenesis might also contribute to tumor progression, if not drive it. So to investigate angiogenesis in vivo, we used dynamic contrast enhancement MRI, which allow semi-quantitative evaluation of the perfusion of a contrast agent over time. And the temporal changes in signal enhancement measurements reflect the functionality of the vascular system. So here you can see the distribution and the concentration of the gadolinium at the T0 and the T of maximum intensity acquisition. And we can see that the representative SH-controlled tumors show a large peritumoral perfusion, where SH-LRP1 tumor shows a reduced perfusion to a peripheral part of the tumor, allowing us to conclude that the perfusion is more superficial in SH-LRP1 tumors. Then the microvessel density after a CD41 immunostaining revealed a decreased vessel number in SH-LRP1 tumors. And more than that, we could observe a quite different vascular morphology and organization in SH-LRP1 tumors. As you can see, most SH-LRP1 CD31 stained tumor section exhibited large structures looking like hemorrhagic lakes rather than vessels, but also excessive anastomosis were observed. So this highlights a sustained but anarchic and incomplete stimulation of angiogenesis when LRP1 is repressed in MDA-MB231 tumors. We then looked at hypoxia and necrosis within the tumor. In vivo, thanks to the use of the hypoxysen 680 probes in FMT, which allowed us the detection of the carbonic anhydrase 9, we have demonstrated a significant increase in hypoxia in SH-LRP1 tumors. And this increase in hypoxia have been correlated with an increase in necrotic areas revealed by a basic HE staining. And these results confirm our previous regarding the role of LRP1 receptor on tumor vascular functionality. So then, to focus on the role of LRP1 in tumor angiogenesis, we used a matrigel plug model to evaluate the formation of blood vessels in vivo in a gel mimicking a matrix environment implanted subcutaneously into mice. So basically, we injected SH control or SH-LRP1 cells mixed with matrigel into mice. At the end of the protocol, the tumor were excised and analyzed using a CD31 staining. And as you can see, immunohistological analysis showed a decrease in the number of vessels in SH-LRP1 plug compared to SH control. During the experiment, we used different imaging modalities to extract functional information about the developing vascular network. So using DCA MRA, we analyzed the area under the curve of the first seconds of perfusion. And as you can see, the speed of the filling phase, as well as the maximum intensity after the filling phase, is more important in SH control than in SH-LRP1, allowing us to say that the perfusion is less effective in SH-LRP1 matrigel plug compared to SH control. In agreement with these results, we demonstrated using the Angiogenesis 680 probes a decrease in vascular density of 86%. Altogether, these results emphasize the importance of the tumoral LRP1 receptor in the formation of blood vessels. So this second part aims to characterize the effects of the secretome of LRP1 repressed or not MDA-MB231 cells on some major functions of endothelial cells, such as pseudotube formation and migration. In pseudotube formation assays, we could observe that when UVECs were incubated in SH-LRP1 TCM, their ability to organize themselves into tubular structure was significantly reduced compared to UVECs in SH-controlled TCM. Then, using transfer system for migration assay, we evaluated the chemoattractant effect of the TCM on UVECs migration through an 8 micrometer porous membrane coated with fibronectin for 12 hours. The number of migrating UVECs toward SH-LRP1 TCM was decreased by 58% compared to SH control. Considering this, LRP1 repression in MDA-MB231 TCM appears to lower the angiogenic potential of endothelial cells. Since angiogenesis is also based on the ability of the cells to degrade, reshape the matrix, we measured plasmin activity using a commercial plasmin substrate. First, we showed a decreased plasmin activity, or we can also say a slower conversion of plasminogen to plasmin in SHLRP1 TCM compared to SH control. And then, using conditioned media prepared from UVEX previously treated with SHLRP1 or SH control TCM, we showed an even important difference in plasmin activity. And this result suggests the potential presence of plasmin inhibitors or its activators inhibitors in the secretome of SHLRP1 cells and UVEX cells previously treated with SHLRP1 TCM. So in this next part, we try and identify molecular actors involved which tip the balance toward a decrease in tumor angiogenesis when LRP1 is repressed, using proteomics experiments that were carried out by the OncoProt platform of the University of Bordeaux in France. From this data, a heat map was generated and allowed us to visualize rather quickly that the secretome of the SHLRP1 cells, the 3 replicates on the right, is largely enriched compared to the SH control. Then, to allow a better visualization of its data and to highlight the signaling pathway potentially involved, we end up with a network representation of the proteins, which revolved around 2 pathways, the TGF-beta and the plasminogen-plasmin system. So we deliberately add the LRP1 receptor in the center to make the link with the protein found. And indeed, we can see the enrichment of proteins that interact directly with LRP1. Among the targets identified are proteins that support a strong regulation of proteinase activity, which support the results obtained regarding the decrease in plasmin activity when LRP1 is repressed. We also find many proteins that support the anti-angiogenic effect that we observed, such as TIMS or thrombospondin 1. But as you know, angiogenesis is controlled by the balance between pro and anti-angiogenic environmental signals. So it's not surprising to find an enrichment of pro-angiogenic proteins in the secretome of SHLRP1 also. The idea is, therefore, from the data we have, to focus on some targets which appear relevant insofar as they constitute the argument of choice in the anti-angiogenic effects observed when LRP1 is repressed, such as TIMP, notably TIMP3, but also thrombospondin or also actors of the plasminogen-plasmin system. So to summarize, we have shown that LRP1 supports tumor growth in vivo in a TNBC model and acts as an important player in maintaining a certain tumor vascular homeostasis, which is necessary for tumor growth. In vitro, using tumor conditioned media, we have shown that LRP1 expression in tumor cells directly influence the angiogenic potential of UVEX. And finally, we showed that LRP1, due to its multiple roles, modulates a number of factors which impact the overall angiogenic balance, reflecting the complex regulation of tumor angiogenesis in vivo. The accumulation of all this data allowed us to propose a model of what the secretome of SHLRP1 cells might look like with the various proteins identified that could play a determining role in the observed effects. So researchers in my team are currently working to validate these results by using other TNBC models, but also other models such as the MINT model, which allows the different stages of tumorigenesis in a single model. But the data we have led us to the following question: what role might LRP1 play in the late stages of tumorigenesis? So previous and preliminary results show that the repression of LRP1 in MDA-MB231 influences the invasive capacities of cells in vitro according to hypoxia, which led us to the hypothesis that LRP1 could potentially have a bivalent role in the late phases of TNBC, especially in response to hypoxia. So to go further, researchers in my university are now interested in the role of LRP1 in EMT in view of the enrichment of the TGF-beta pathway, but also in the role of LRP1 in cancer stem cells enrichment to try to decipher the role that this receptor could play in the formation of metastasis and the relapse of the disease. Thus, in view of the pivotal role of LRP1 receptor and its many interaction with the pericellular environment, its specific targeting does not appear to be a strategy of choice in the treatment of TNBC, but understanding its role reveals a source of many orientations to deepen. I would like to thank the organization for allowing me to speak, for allowing me to present my work with you today. I want to thank all the members of the medic unit research, as well as all facilities that contribute to this work. And of course, our financial support, La Ligue contre le Cancer, and the industrial MR Solutions. And thank you for your attention.